Hi everyone, welcome to the online Causal first seminar of the new academic year. We're excited to kick off the new year with a seminar given by two brilliant young researchers. Our first speaker is Jiaqi Zhang from MIT. She will be talking about learning causal cellular programs from large-scale perturbations. And our second speaker will be Siju Liu from UC Berkeley. She will be talking about estimating treatment effects with competing intercurrent events in randomized control trials. So the questions today will be handled by Sarah. Sarah, please go ahead to introduce that.
Thank you. This is just to remind you to please use the question and answering tool on Zoom instead of the chat. I will be asking the questions after each talk. So after Jiaqi will be answering the questions. So asking the questions and Jiaqi will be answering the questions. And after Siju, I'll be also asking questions. But Siju also has one of her co-authors, so he will be answering questions already during the talk. So, okay, without further ado, I will let Jiaqi start her talk.
Thanks for the introduction. Thanks for inviting me. I'm gonna share my slides. Can you all see the slides? Cool.
Yes.
So hi everyone, I'm Jiaqi. I'm a final year PhD student, and I'm gonna talk about learning causal cellular programs from large-scale perturbations. So the human body is composed of many different cell types. For example, there are muscle cells, blood cells, also nerve cells, and they perform different functions within our body, but they kind of all originate from this embryonic stem cells. So this raised kind of a fundamental question, which is how do we actually generate such a diversity of cell types during development? And also, even within a single cell type, cells can exhibit different functional states and respond differently to disease. For example, take T cells. They are different kind of cell states, including progenitor effector and terminal exhausted. The progenitor cells, they tend to be more effective in terms of immune responses, whereas terminal exhausted cells could be weaker in terms of tumor killing capabilities. Studies have suggested that it's possible that we can shift the cell state using external perturbations such as genetic or chemical stimulations. But the question really is, how do we actually design interventions such that we can achieve such desirable outcomes? So from these motivations, we can identify two key problems. First of all, to understand these complex cellular systems, we kind of need a conceptual or theoretical tool that can explain the observed behaviors to guide our biological discovery. And the second problem is to be able to engineer the system, we actually need models that will allow us to predict the effect of perturbations and maybe generalize to predict the effects of novel perturbations that we haven't tested yet. So these are all kind of very hard questions, but fortunately, we have access to a very unique hammer in biomedicine, which is the ability to perform large-scale perturbations. So recent advances in biotechnologies have allowed us to systematically perturb cells and also measure their response. For example, in single-cell genomics, we can perform genetic perturbations using a technology called CRISPR. This will allow us to go into the cells and actually just knock out or overexpress specific genes. And then we can observe their effects in thousands or even millions of single cells. So there are multiple ways we can kind of measure these effects. On the top, we have this gene expression matrix. This corresponds to a type of experiment called perturb-seq, which allow you to measure gene expressions in single cells, where the rows correspond to cells and the columns correspond to genes. And then you can assign each cell into individual perturbation environment. Whereas the optical pool screen at the bottom, this sort of capture phenotypes such as cellular morphology or protein localizations. And the current style that we can do this experiment is very remarkable. For example, genome-wide screen, perturb-seq screen from 2022, it has over 9,000 genes perturbed in millions of cells. So this kind of provides us a very unique opportunity to poke at the underlying mechanism. Just to show a very simple example of how it might help. we're probably very familiar with this very simple 3-node example. If we observe 3 variables and they're associated in this way, we know that in observational cases, if we don't assume additional information and there's not a V structure, we cannot really direct the edges. But by perturbing like certain nodes, for example, Z2, we would know that all the variables that actually are changed from the observational case would be downstream of Z2, so we can direct the edges. So this is a very simple illustrative example of seeing how intervention might help us poke at the underlying mechanism. So now we have this very powerful hammer of large-scale perturbations. We can look at the first problem, which is how should we really understand the causal programs that drive cellular behavior? So traditionally, people have represented causality using a directed graph like this. So this is behind a very specific process in sea urchins, where each node variable here corresponds to a gene, and then the edges reflect regulatory relationships. However, I would like to point out that learning such a full causal graph can be extremely difficult. It might require like a significant amount of prior knowledge of how the system works. And then based on those prior knowledge, you would develop hypotheses, which then lead to small local perturbations to test this hypothesis. But then if we look from, say, a data perspective, where we have already collected a relatively unbiased large screen, we might ask, what should really be the causal variable that can explain this unbiased large screen or data? Should we treat individual kind of dimensions here as causal variables? Oftentimes, probably not. For example, in the expression matrix on the left, we would see that a lot of the variables, a lot of the columns, they kind of move up and down together, or in other words, they co-express, suggesting that probably we should really group them as one individual causal node. And then on the right is more obvious, where individual dimension here are pixels, which shouldn't correspond to meaningful causal variables. And we kind of need to aggregate them and collectively represent, say, for example, a cellular shape or something like that as one variable. So this leads into the proposal here, which let's take the expression matrix for example. Instead of treating individual observed variables, in this case, as genes, as causal variables, we're going to aggregate several dimensions here together as one causal variable and learn it as the latent representation behind the observed data. So intuitively, we're following the principle that we're going to aggregate variables, genes here, such that they will have similar effects if you perturb or intervene on them. And they will also have similar responses on their different interventions. So this has been studied, learning latent variables has been studied quite extensively in ML and statistics under different frameworks like ICA, disentanglement, and etc. But oftentimes, the latent factors are assumed to be independent, which is not really the case here, because we know that these factors, in this case, programs of genes, they should really be causally related. Therefore, the question is, how do we resolve the latent factors and the relationship if we only have them after this unknown mixing? So we studied this problem, and then the first question we ask is identifiability, which is essentially asking if we have a data generating process like this with enough data, is the latent model that explains my data unique? Or in other words, if I have the latent variables U that generate the observed variables, could I uniquely learn from the observed variables a U hat that equals to U? So our results shows that with enough samples and suitable assumptions, namely, we have to assume the mixing function follow a specific form, a faithfulness of the interventions, such that they don't degenerate to observational cases, as well as enough interventions, namely at least one intervention per latent node. Then the latent intervention targets of these interventions, as well as the causal structure between them, are identifiable up to permuting the variables. So we can tell from the interventional data which are the interventions that are targeting the same latent variable and how are the structures between this latent variable look like. So just to provide very brief intuition of how we get to such identifiability result, the first step that we did in our proof is sort of resolving to this linear identifiability of U, which namely we're going to identify U hat that we're going to show equals to U times the matrix gamma. And then since we know that the latent variables U, they are sort of follow a topological order, we're going to enforce this order so that this will make sure that the gamma matrix up there in step 1 is going to be upper triangular. And then afterwards, we're going to enforce that there's sparsity in U hat when we're changing, when we're performing perturbations. This will then allow us to have a structure in gamma that retrieve the ground truth causal structure. So with this, let's look briefly at how we can design an algorithm to learn them with data. Here's kind of the data that we have. We have like a group of control samples where no interventions are applied, and also a group of perturb intervened samples. So to model the changes here, we can take any feed-forward network, and just, for example, conditional VAE, feeding a control sample, a perturbation label, and generate an intervened version of the cell. But it's kind of a problem here because the observation data and intervention data, they are not paired. They're not counterfactual pairs. So we kind of need to introduce this discrepancy loss in here, which essentially means we're going to generate we're going to feed in a group of control cells, and generate a group of intervened cells, and then just use the distribution of loss between them to train a model. So we don't really know the individual pairs, but we know that distribution they should look alike. And this will actually allow us to learn the map from control to intervened distribution. We can also easily build in the component to learn the latent causal variables here by just enforcing that the latent variables, we first have U and then the intervention I, and then enforcing I only changes U follow a structure. And then this, we can get an intervened version of U that reflects the causal graph. So when we learn them jointly, we can actually interpret the latent U as the causal variable and the graph between them as the causal relationship between U. So in theory, we can show that this algorithm actually provably identify the latent causal variables and the structure between them if we assume infinite data zero training loss and correct prior specification in the variational autoencoder. So it actually achieves the general identifiability given before. So with this, we kind of want to validate it empirically using an actual data. So we took a perturbation gene expression data. This one has roughly 100 single gene perturbations and 100 double gene perturbations. We only use the single gene perturbations, and we apply our algorithm to learn an abstracted causal graph between the programs of genes. Here's a representation of that graph, where each node we're representing it using a representative genes. So I'd like to point out that a certain relationship here has been studied before. For example, the stage between DUSP9 and MAPK1 ETS2 was actually identified in the original paper. So it provides us some sanity check that we might be able to pick up actual relationships. It also gives us some confidence to investigate other hypotheses generated by such methods. We also showed in our paper that if we're actually able to achieve the identifiability results shown previously, we could also, in theory, extrapolate to predict the effect of combinatorial, in this case, double gene interventions. If those interventions follow the assumption of they change independent causal mechanism, and then it follows the structure causal equation to define them. Take an example, if we have two individual perturbations targeting U2 and U3 here, what we're going to do is we're going to take their effects and then just combine their learned effect to the nodes that are downstream of both and add their effects for the variables that are downstream of both. So here are 3 examples of applying this approach. These 3 are UMAP plots showing 3 types of combinatorial perturbations where you see a lot of dots. Each dots correspond to a cell projected down to 2 dimensions. And then the blue dots are the actual cells, whereas the orange ones are the generated cells. And then the background gray is the manifold of all other cells. So we see that for the two cases on the left, KLF1 plus EBPA and this middle one, we can actually predict their effects, but whereas this other on the right, we cannot predict effect because there might be interactions between their perturbation effect, and we can't really use the previous definition of combinatorial intervention to extrapolate to their effect. So in general, we can sort of use this approach to identify interactions of interventions. And then this might not be exactly the same as if you were to define it in the original observational space, because you're defining interactions within the latent space. For example, this one on the lab where we call it additive is actually epistasis in the original paper. But then if we're actually able to predict it, their interactions shouldn't really be very non-additive. So I'm going to move on to the next question, which is, now it's possible that we can generalize to combinatorial perturbations. How about novel perturbations on new targets? So previously, we were encoding each genetic perturbation using a one-hot vector, and the perturbation encoder will learn how to map the perturbations that belong to the same program into change of the same latent node. But however, if we use one-hot vector here to represent, say, a novel perturbation, we're gonna be facing a scenario where in testing, we're gonna have a one-hot vector on an entry that was never seen during training. So this is going to result in a very arbitrary prediction. So we can't really do this. What can we do? So we thought of a way to resolve this. Is quite intuitive. Since we have a lot of prior biological knowledge of these genes, how about we just use this knowledge to, instead of the one hot vector, to represent the genetic perturbation here. So this way, if we use the prior knowledge, we can relate unseen perturbations to seen ones, as they would have been observed in prior literature to have similar functions. So this leads to our model, which we call MORPH, a modular framework for predicting response to perturbation changes. Modular being that each component here, you can swap it out with alternate different kind of options. And then we apply this to predict the effect of unseen interventions. Just to make clear of the setup, we have a set of perturbations for training. And the set of perturbations, we're going to encode their perturbations using a prior biological knowledge. And there are various different prior biological knowledge that we can use. For example, we can use textual embeddings from GPT about these genes, or we can use a previous relatively cheaper modality measuring perturbations in different cell lines in the DepMap project, or we can use gene embeddings that were learned from previously trained single cell foundation models. So once we represent the genes using this prior knowledge and learn this network, we can then apply it to unseen perturbations to predict their effects. And it's going to relate the unseen perturbations to the seen ones because they might have a similar representations based on the prior knowledge. So here's a snapshot of the result on a cell line called K562, which here roughly is the genome widescreen targeting essential genes. It has roughly 2,000 number of perturbations. And we can see that our model MORPH, which is in green, compared to the state-of-the-art baseline, which in fact is actually a linear baseline, is outperforming all these baselines in terms of various different metrics. MMD is measuring distribution changes, and these two are MMC and Pearson correlation. Here's also a very illustrated example of the prediction where we say similarly has the UMAP as before, where dots represent cells. And then here, the red one corresponds to the actual perturbed cell, and the blue ones correspond to predicted ones. We can see that on the right is our prediction, which corresponds to, like, we get the distribution quite right in this case. And we also saw that using different priors, they can lead to different results overall. We see that this result comparing different priors, also we use the gene former gene embedding as prior as well, and control gene expression is just a baseline, standard baseline here. MMD is lower the better, RMSE lower the better, and Pearson correlation higher the better. we see that that map here is dominating the performance. It's actually the only prior that actually directly measured the experimental perturbation data. However, it's in a much cheaper modality, measuring only viability instead of the whole gene expression matrix. This kind of highlights the use of functional grounded information to improve generalization. And also, if we were to break down the performance to, like, say, individual sets of perturbations, we see that the comparison between different approaches, for example, gene PT is actually not doing so bad on the ribosomal subunit, as well as maturation of SSU rRNA, suggesting that there are kind of different information in the prior screen for the perturbation effects. Which is quite interesting. So this shows promising results of predicting unseen perturbations, which can be applicable to a variety of tasks. In the papers, we investigated many of this, which I'm not going to give in detail here. If you're interested, our paper is on bioRxiv. You can read about it there. However, just want to highlight one last point on this. Since the overall framework is modular, essentially we don't require the data modality to be very specifically to gene expression or tabular data like that. We can apply it to different modalities such as optical screens that are images of cells. So we took a look at a genome-wide screen on Ebola virus, and we actually just extracted image features from it and then tried to predict based on a group of seen perturbations, see if we can predict for an unseen perturbation whether the images of the cell will have a corresponding measured cell state in their condition. And then we see that our method in the green here is outperforming, in the green line here is outperforming overall to the state-of-the-art methods here, suggesting that incorporating this prior in this way is actually very helpful. So with that, I will conclude my talk today. Essentially, I gave out two types of ideas that we did. The first one is how do we define and learn causal programs between high-dimensional or perceptual data that we have before. We're suggesting kind of aggregating variables such as genes where they have similar effects or responses to perturbations. That's one latent variable. And then secondly, I talked about an idea to model the perturbation effects. We can have this modular discrepancy-based models to measure the changes on the distribution level. And we suggest the combination with actual tons of prior data will be helpful for us to generalize to predict an unseen perturbation. There are several extensions we can think about over this to work. The first of all, in the causal representation learning framework, we kind of have those various assumptions as including mixing function as well as number of interventions. So the question really is there a way to relax the assumptions there? There have been many follow-up work that resolve more difficult situation. We actually also did one paper where we only look at the cases where we can identify from our observational data. And the second extension is in terms of modeling component. In the previous cases, we're actually treating cells as IAD samples. So this is not going to be effective if we actually consider a tissue environment where we have the cells neighboring each other. And they might communicate with each other, which introduce interference between them. So how can we actually model this? And the second case being that if the system is actually dynamic and we have time series measurements, how do we model that? And then finally, how do disparate approaches actually perform in an experimental design setting where we're doing actual experiments? So hopefully I can talk about the experimental design in a future iteration when I have more time and more results. And here are the two references for the two works that I introduced today. And I would like to thank my mentor, Caroline, as well as my amazing collaborators between these two projects. And thank you guys all for listening.
Okay, thank you, Jiaqi, for your talk. There are already some questions, so I'll start asking them. So Brad Ulko asks, are priors as introduced soft probabilistic bias or hard priors, sorry.
I would say more or less hard biases because we're just encoding them as a vector. And although you actually have a perturbation encoder that can change the representations, and then that after the perturbation encoder, you get a representation that reflects a combination of the prior as well as all your training perturbation data. And that will reflect some changes to the embeddings of the perturbations.
Okay. Okay, so just a second, I'm going to ask a clarifying question to another answer. So I'll ask you, in the meantime, I'm waiting for a clarification on a question. I wanted to ask you something else. So I was curious about interventions that you're using. So what about if instead of having interventions, maybe you have already thought about it, but what if you have distribution shifts because you have different environments between the cells? Could you use the methods you developed in this setting, or have you tried?
It's a very good question. I actually tried it. Let me see if I can go to your previous slide. Something closer to that. See, the second kind of task that we apply it to is called transferring perturbation effects. So this is actually taking like one cellular context and try to see if we can use results there to predict in another cellular context, which kind of is a different cell line. They will have distribution changes there. And then we saw that you're able to do it if your perturbation effect, they kind of align. So if a perturbation will do two types of things in those two contexts, like drastically different things, then you won't be able to transfer the effects. But in another scenario where they're kind of similar, then you can do it.
Okay, makes sense. So there is another question on priors.
Yeah.
The question is, were the priors used as a single latent variable or used as a condition, like a conditional VAE?
Condition as conditional VAE.
Okay, makes sense. Okay, I'm going to ask you maybe just shortly another question. I was also curious about the fact that you mentioned, but maybe I misunderstood, that at some point you're selecting the single interventions in some of your work. And I was wondering, why didn't you select also the ones which have multiple interventions, multiple targets?
I see. Yeah, I think previously the one maybe you are referring to this example where we were applying. Right.
Yeah, exactly.
Yeah, the reason is in the theorem of the proof, we were only dealing with the case where we have single node interventions. So the theorem is saying that, yeah, exactly. And then that you can identify it. And then we're holding out the double ones because we have another theorem that says if you're able to resolve it, then you can predict using none of the combinatorial data. So we're holding that out. But if your goal is actually just to learn as much as about the regulatory information as possible, then you should use all the data.
Okay, fair enough. Okay, then there is a question that I'm not sure exactly what it refers to. It's about interference, and it says what to do in case of TE interference.
I actually don't have a good answer for that because that's the one probably referring to one of the extensions that I mentioned here. So maybe come back to that question in a few months.
Okay, fair enough. Okay, so I think it's now time to kind of conclude. So thank you, Jiaqi. And now we're going to move to Siju.
That's good. Stop sharing.
Thank you. Yeah. Okay.
Okay, I'm gonna share my slides first.